the endomembrane system. So the end part here is internal. The internal membranes are going to be a distinguishing characteristics of eukaryotic cells, the more complicated cells. Parts of the endomembrane system, we're going to look at 6 of them. The first one is the nuclear envelope. And the nuclear envelope is right here. As you can see, it's a double membrane. It has pores, so things can get in and out, but it's regulated what can come in and out. The next will be the endoplasmic reticulum. My handwriting. This is abbreviated ER, and there are 2 types of ER. There's smooth ER and rough ER. Rough ER is studded with ribosomes, and that's what makes it rough. There's a Golgi apparatus. Which is here. This is the name, and then it's the stacks of kind of flattened, they look kind of like pancakes, flattened on top of each other. We've got lysosomes. Here's a lysosome. We have several types of vesicles. A vesicle, this one is a transport vesicle, and here's another transport vesicle. We also have vacuoles. This is a plant, an animal cell, and so it doesn't have a vacuole. Vacuoles are found in plants. So let's look and see how all this works together. Say we want to make a secretory protein. We're going to, this is a cell that's going to make this, and it's for export. The ribosome that's bound to the ER, endoplasmic reticulum, manufactures the protein. And we'll find out later, ribosomes make protein. And so it excretes our protein and puts it in this space right here. Then, sugars, short chains of sugars can be added. These can be added any place along the way. That leaves the ER in a transport vesicle. So now our protein is inside that transport vesicle. Our transport vesicle will fuse with this side of the Golgi apparatus. The proteins may be modified as they pass through these stacks. And then eventually, they will be put in another vesicle and excreted. So let's see, let's go fill in our spots here. It fuses with the Golgi apparatus. Modified in short term. So this is kind of, you can think of it as a warehouse. I've also heard post office. That things come in and they get sorted and shift around, and then they get shipped off again. Then they're modified and they fuse with the cell membranes. Before we go on and look at this table, I want to go to the PowerPoint and look at some other vesicles that may be formed. This is the lysozyme, and it's got some digestive enzymes in it. And this is the cell in here, and this is outside the cell. So this is some food or other particles that we want to take into the cell. So the plasma membrane will bow in and take some of the food and make a vacuole. So now we've got food in here, and it's inside the cell. The digestive enzymes from the lysozyme will join. So these two will join each other, and then the digestive enzymes can digest the food. So that's another way the endomembrane system can work. Let's go back to our... Okay, so let's look at a couple of different components, some other components. So we've got lysosomes. We just saw that these are digestive. And they can break down food, or they can break down components of the cell for recycling. If you have an old mitochondria that's not working very well anymore, you can digest it down and reuse its components. Vacuoles have all sorts of different uses. They are used to expel water. They can be storage. They can store protein, for example, in seeds. They can store pigment. If you're a flower and you need to store pigment so that you'll be a pretty color. And they can also store waste. Vesicles and vacuoles are plants. Vesicles can transport proteins. That's what we saw in this diagram above. We had transport vesicles. And they can transport from the ER to the Golgi apparatus, and then to the plasma membrane. So ER is endoplasmic reticulum, GA is Golgi apparatus, and PM is plasma membrane.